Okay, I'd like to introduce you to Dr. Mark Stibich, Carroll High School class of 1991. And he is the Chief Scientific Officer, Epidemiologist for Xenix Disinfection Services. Am I saying that correct, Mark?
Yes, perfect.
Okay, so I'm gonna bring it over to you.
All right. And let's see. So first I want to ask, is it John and Max, what's useful to you today? What would you like to hear about? I could talk about anything.
Just go.
All right, I'll just go. So I will give a quick overview of what I did since Carroll, if that's helpful, and then we could go into any sort of specifics. So first I went to undergraduate here at Yale, and I majored in something completely useless. I was interested in philosophy and Buddhism, and that's what I majored in at Yale. So after Yale, I went to Peace Corps in Mongolia, and that was a really great experience for me. So I learned Mongolian. I lived there for 2.5 years. There's a little, down here you can see the little, they're called girs, or they're also called yurts, the round felt tents. I lived there, and in there they were starting a new program. This was, we were the 7th Mongolia group, and they're starting a new program. And one of the things they were looking for is trying to start a health program. And they asked for people who wanted to do that. And I had an EMT at the time, a wilderness EMT, so I volunteered. And that actually, of all the things that kind of set my direction, that was one of them, because I really realized what the health system was like. And it wasn't so much the education of the doctors, it was really these public health system things. So that led me down this path to this school, which was the Johns Hopkins School of Public Health. And so from Peace Corps, I went there and I got my master's degree and stayed on for my PhD, and really got interested in infections and how infections kind of spread in communities. And what's really interesting is when you start looking at the time I was, I started getting involved in HIV. And you start looking at, you can go in public health, you go everywhere from like the teeny tiny molecular pathways of, the molecular immunology pathways, and try to understand how the illness is working there. And you go all the way up to policy, because like government policies and the way we handle vulnerable populations and drug availability, treatment availability, also very much impact an infectious disease. And we're seeing that so much now with COVID is, so for me, public health is really interesting because you could kind of do one zone and you could jump to another zone. So for example, with COVID, we could be examining the molecular immunological vaccine drug development pathways. You could go in that direction. But obviously, policy makes such a huge impact on the course of the disease in our country. And public health is sort of branching all of that. And so you have to have knowledge in all of those different areas and be able to work in there. But depending on what your goals are and what program you're working with, you may jump in those areas. So I've done some molecular stuff. I've done a lot of microbiology, and I've done policy work, and we started a company. So I'm a big proponent of the public health field, because you really can approach it in whatever area is interesting to you. And you can also jump from one thing to another. You can work in one zone for a while, become a content expert there, but then you could jump onto another project. There's just seems to be a lot of flexibility. And so public health is I'm a big fan. A place I went after Hopkins, it started in Hopkins, then afterwards was this organization, which is very fun. This is called Speed Infoshares, or AIDS Infoshare. It's a Russian organization. And basically they did, and still are doing, extensive infection prevention in HIV in Russia and the former Soviet Union. So I got very involved with that organization for a while. And there was, at the time, this was the late 90s, there was a lot of funding available for work. But with Putin coming and the politics and everything else, it was looking more and more that American outsiders working with the Russian organization was not going to be sustainable. So we came across this technology, and that is what led to this company, which is Xenix. And so this is where I spend most of my time now. About 10 years ago, we started this company, and what it does, it's a germ zapping robot. So we're not germ zapping solutions because we have more products, but about, a month ago, earlier in the year, we were germ zapping robots. And what we have, and it should pop up here, there it is. So it's this device. And what it has inside of it is a pulsed xenon lamp. So when you apply high voltage electricity to the lamp, it produces essentially a plasma. Essentially, you're making lightning in that lamp. And when you do that energy, so you put 35,000 volts of energy onto that lamp, that energy has to be released, and it's released as heat and light. And when you do it very, very fast, it's reduced as high energy light. So as you look at the electromagnetic spectrum, you have infrared on one end, you have UV on this end, and that's the light we're exposed to, and then you go into X-rays and everything else, radio waves. What this produces when you get the engineering right, you produce something called UVC, or germicidal light. And so this is light that is, we know UVA, UVB, sunlight, they do damage to our cells, our epidermis. UVC is filtered by the ozone layer. So it doesn't naturally occur on Earth, which means bacteria and viruses over their 4 billion years of evolution haven't been exposed to this and haven't developed defenses against this type of light. So it's a high energy UV. And basically it'll go in and it will fuse with the DNA of organisms. And I think we have, I don't know, I haven't been on my website for a while, so I'm not sure where, we just redid it, so I'm not sure where everything is. Not there, not probably here. And if you go here, we took it all down. Okay, we took a lot of stuff down recently. But what you see is, you see the DNA of an organism, or the RNA of an organism, and you know how there's sort of the vertical connections between the acids. What the photon will come in and will disrupt that connection and basically make it bond to another thymine. So you get a thymine to thymine bond, which is called a thymine dimer. That's one mechanism. Other mechanisms that will disrupt the actual phosphorus chain, the backbone of the DNA, and split that. It will also insert water molecules into the DNA, and will also lyse cells. So it'll actually damage the lipids and proteins that form the cell wall, mostly the lipids, and cause the cell wall to burst open. So it's a very good disinfectant. But what's nice about it is it also works against viruses, and it works against SARS-CoV-2. And so early on in the pandemic, we did some direct testing against SARS-CoV-2 in it was actually a BSL-4 lab at the time in Texas. So we were able to show that we were able to get 99.9% disinfection, deactivation of the SARS-CoV-2 virus. So we've been around, this company, we've been around 10 years. We've disinfected more than 25 million rooms in something like 22 countries. And we now have about 700 hospital customers. And it's based in San Antonio, Texas, is where most of and we do all the manufacturing ourselves. And now we're moving into these other areas. My role with this has been, so I'm an inventor, I'm the chief scientific officer. Most of the team, is building robots, keep maintaining robots, working with customer sales, finance, accounting, all of the normal business functions. I get the fun of being able to develop new products, look at new ideas, work with our customers on research. And so we've had a bunch of research studies come out. So this is one that we're particularly proud of. So this is the Mayo Clinic reporting a reduction in their C. diff infection rates after they used, this is before, this is in February, but this is before the pandemic. And basically what's happening in the hospitals right now, when someone goes into a hospital, it's likely they have their immune system isn't 100%. They're sick. And then if they're exposed to organisms, pathogens in that hospital environment, the risk of them developing an infection is increased. So these are called healthcare associated infections, or HAIs. And they affect at least 2 million people a year in the country. They cost roughly $50 billion for the healthcare system to deal with these infections. Because what will happen is someone goes in for, something, a simple, maybe they have a wound or something like that, and then they develop an MRSA or a MRSA infection. They might be in the hospital 14 days when they possibly shouldn't have been in the hospital at all. And that additional care is now the hospitals have to pay for it. If they cause the infections, the hospitals have to pay for it. So it's a huge problem. And what we're able to do is remove the risk that the environment causes to that problem. So to the degree the surfaces are contaminated with these pathogens, we're able to come in and in 5 minutes we can disinfect a room. So it's really something that can be operational, something that fits into hospitals. Hospitals are really busy, chaotic places. So we had to come up with a technology that not only was effective in reducing the organisms, but was also fast enough that we weren't holding up patients, because the hospital can hold a room for 2, 3, 4 hours to clean it because people are waiting in the emergency room. And the hospitals operate at that level. They're always right on the edge of being, overstaffed. So there's always a little bit of a wait time. And so we had to come up with a technology that could be easily used by the housekeepers, but was effective. And so this study shows C. diff, which is called Clostridioides difficile. It's a bacterium, and in the environment, it forms a spore. So it's called an anaerobic bacteria. In the presence of oxygen, it doesn't like oxygen, and it forms a spore. So it forms basically a hard shell around itself. When it does that, it becomes fairly impervious to almost all of the regular disinfectants. So the normal kind of nicer disinfectants that hospitals use, quaternary ammonia, those type of disinfectants, they don't work against C. diff. And C. diff can survive in the environment for up to 6 months. So once you have a patient with C. diff, and it causes this just horrendous diarrhea, and it's the #1 sort of hospital infection right now. And so, as you can imagine, with horrendous diarrhea, you have a truly vast spread of the organism in the bathroom and the area around it. But it's very difficult to remove. You have to use bleach, like alcohol, things like that won't remove it. So you really either have to do a very thorough manual bleach disinfection or use something like a disinfecting light. And what we found is in hospitals, when there's been these great studies, they'll go out and they'll mark surfaces with a black light dye, a fluorescent dye. So you can't see it. And then the housekeepers come in and they clean the surfaces. And then all they have to do is wipe it to remove that dye. And then after the housekeepers come in, you take your black light and you go see which surfaces still have the dye on it. And you can tell if they cleaned them at all. And typically over studies of 10,000 plus rooms, housekeepers clean about half of the high-touch surfaces. So if you mark a tray table or a bed rail or a light switch, they'll clean about 50% of them. Because and that's never, I don't really think that's their fault. They're given sometimes they're given as little as 18 minutes to come into a hospital room after a patient's been there, to take out the trash, to clean the floor, to potentially remove blood on the floor, towels, everything that's there, trash, everything that's around, wipe everything down. And then also somehow they're supposed to disinfect the room too. It's just not humanly possible. And that's really the gap we saw with this technology. And we can come in and we can automate that process and remove the human error. So that's what I do most of the time. So, so as we go out to different areas, we need to figure out protocols. How do we use it from an ICU room to an operating room to other places? And then recently we're moving into more non-healthcare areas. So this was just released last week. So we're disinfecting the Panthers, the Carolina Panthers football team. So they're using us to disinfect their locker rooms and all of the player areas. But if you picture just like even a gym, like you picture all of the different equipment, to go with the wipe and manually disinfect all of that equipment is nearly impossible to do correctly. But a light works really well. So we're moving into these other areas, and my role is to figure out, okay, what's the right use of the robot in these areas? And then, so a couple things we've done that I'm kind of proud of is we have, it says 37, we actually have 43 publications. So this is PubMed. So PubMed is an amazing database of all of the medical literature. So you can just go on PubMed and search anything. And these are all studies about the Xenix robot. So we have studies that look at, here's one in a Japanese hospital that looks at, the amount of contamination before and after, a couple Japanese studies. We've got, using this one, I don't know if it'll come up if I click on it, but this one, we created a pod. Let's see, it should be on here. Like again, I apologize because we have changed the website and I'm not familiar with it anymore. Here we are at the San Antonio airport. But we created a pod. So the pod is basically a pop-up tent. So you could bring equipment into that tent and then use it to disinfect. And then this study, was on how the disinfection of things in the pod. And what you find is, not surprisingly, is when you take a wheelchair and it goes through its normal manual disinfection process, and then you sample it for organisms, it's actually quite contaminated still. And then, so it needs another layer, another method of disinfection. And we've got, stories, Ecuador, we've got this fungus, Candida auris was a big pathogen showing up last fall. It's very difficult to kill, very difficult to remove from the environment. We have, it's not listed here yet, but we have our SARS-CoV-2 data. So this is one thing I work on. The other thing I'm very involved in is this side. So this is Google Patents. And we have about 120 patents that we filed on the technology or other technologies. And so I've had to learn a lot about how patents work. It's a very strange world. But we've gone in and filed patents, and you file in all these different countries and everything else and try to get, it's its own world of language and how you word things and everything like that. So that's like the quick, I think I'm doing good. So that's the quick overview. I'll pause. You guys have questions? Anything you want to hear about more? Anything interesting?
I have two.
Excellent.
Number one, has there been any organisms that have been shown to resist this at all?
So different organisms will have different levels of susceptibility to UV disinfection. And so you have sort of a hierarchy then of organisms. So some of the organisms that are very easy are susceptible to UV are things like E. coli, what I would call the vegetative bacteria. So these are organisms that when they're in the environment, they're active, they're not in hibernation, they're actively reproducing. Those are going to be more susceptible. And then you have two types of viruses. You have enveloped and non-enveloped viruses, and those have a bit different susceptibility. And then as you go up the hierarchy, you get into things like C. diff, which is dormant in the environment. It forms that spore. And then you go even further up to sort of the ultimate organism to test against is really anthrax. So anthrax is very hardy in the environment. It does form a spore. And that's usually from like EPA or biopreparedness standpoint. That's kind of the gold standard organism to test against. We did test against anthrax. We also tested against Ebola back in like 2014. Ebola is very easy for UV to kill. Anthrax, we are able to kill it. We basically have to run triple or even longer cycles compared to the vegetative bacteria. And then the next level up would be getting into multicellular organisms. So fungus and mold are a bit harder for UV to disinfect against. But that's kind of the hierarchy. SARS falls pretty low on that hierarchy, fairly easy to disinfect. And then again, like anthrax, fungus, mold are harder to disinfect. There are some really strange, they're called extremophiles, organisms that are highly UV resistant, but they're not pathogens, so they don't cause any infections. And the only time that's come up is with NASA. So NASA was interested in using this technology because what they wanted to do was make absolutely certain that they had, if they send the Mars rover to Mars, they want to make absolutely certain they're not bringing any bacteria or DNA or RNA from Earth over, because that would really not only contaminate the planet, but it would also confuse any findings. And my favorite business card I ever got was from this guy. He was at that JPL in NASA, and his title was Director of Planetary Defense. But the planet he was defending was Mars. He was making sure we didn't bring any organisms over to Mars. But those tests were actually quite rigorous because they're looking at these extremophiles that are highly UV resistant. Question one. Question two.
How many jobs do you think this will take away?
So that's a good question. So we emphasize, I don't think any. So, and the reason is, at least in the hospital environment, it still needs an operator. So we have an autonomous version of the robot that will move itself around. But the fact is by the time you get enough power on it, batteries on it, not only is it much more expensive, it's actually not as efficient. So you can't get to as many rooms with that because the robot has to like have a pre-mapped everything. It gets stuck in the hallway. It's really bad at getting going on and off elevators. It's bad at figuring out its sort of plan. And so in the hospital environment, at least, there's always an operator with the robot. So we see it not as job replacement, but as a new tool. And what I like to say because often there's a confusion when we come into a hospital, like the the cleaners, the environmental service workers are nervous that they're going to lose their jobs. And I say, no, what we've done is over 20 years ago, the pathogens really weren't resistant to antibiotics, but the pathogens have evolved. The importance of disinfecting the rooms has become more and more important, but the tools that the cleaners are being given to clean the rooms have not evolved. It's still literally a bucket, a mop, and a wipe. And bleach is a really, really old chemical. And so what we try to say is like, you need a new tool. This is your new tool. So you go make the room look visually clean, make it look perfect, make the bed. You still have to do all of that, but then bring the robot in and let the robot do the disinfection. And while the robot's running, you can move to the next room and get started. I do think in some settings, we're seeing, the autonomous robots, and those are good for like a warehouse or these big areas. And there might be some job replacement. I'm not sure. But so far, I don't think, we actually have hired, some hospitals hire dedicated robot operators, so that creates jobs for them. And we're at probably about 140 or 150 people who are employed with Xenix now, working on it. Good question. Other questions?
What gave you the idea of using light to disinfect them?
So when we were, the long story is when we were here working in Russia, we came across this pulse xenon technology, and it was being used for air disinfection in like tuberculosis clinics. And that kind of didn't make sense because they're bringing it into the clinics after hours to disinfect the air. But for tuberculosis, at least, most of your risk is directly breathing the air of someone who's contaminated while you're in the same room with them. So at night, what settles down, it just wasn't making sense. But the technology was really interesting. So back when I was in graduate school, the idea, there was a lot of alarm bells ringing, but not being really listened to about antimicrobial resistance. Like we were really not using our antibiotics correctly. And there was knowledge that within 10, 15 years, we were going to be using them up and creating these resistant organisms. So I was very, very aware of that problem, saw this technology, and then kind of the leap of faith in some ways was at the time, a lot of the scientific literature was not, or discounted the role of the environment in these infections. They really were not, they really were, yeah, we can clean more, but is it really going to impact the infection rates? Is the environment really creating risk? Which kind of seemed a little crazy to me, but that's where infection control was at the time. They were very focused on sterile processes. So when you insert a catheter, make sure you've done your hands, make sure all your equipment's sterile, make sure you do that process correctly. But they weren't very much focused on the role of the environment in infections. So this was like 10 years ago. And then a couple things changed that. One was a really terrible outbreak of C. diff in the United Kingdom that was linked to poor cleaning of the environment. So they had these outbreaks because they weren't cleaning the environment effectively. So that was kind of in my head at the time. And then we had the technology and we had the problem. And we kind of put it together. And like early in the company, we were in Houston at the time. We just, it was kind of like, we'll take it to the next step and see what happens. It was kind of like, okay, let's just see. So what really kind of solidified it was we did some preliminary testing. We had some models about how fast, we thought we could disinfect a hospital room in maybe 20 minutes or less. That seemed reasonable. And then we went to a conference, and it was in Denver. I remember it so vividly. It was the Association of Professionals in Infection Control. And that year, it was 2008. And that year, the like platinum sponsor of the conference was Steris. And they were launching their room disinfection system. So they had done all their market research and all that. They were launching the system, and they had like a 2-story booth. They were everywhere, and their system, they were estimating to disinfect an OR or a hospital room would take somewhere between 6 and 24 hours. We're like, huh? And at that point, we're like, okay, if they did the research, they launched it, they believe there's a need, they've launched this product, and we have something that's so much better. Like we should really kind of explore this and see where it goes. And interestingly, they pulled that product off the market within a year because 6 to 24 hours, like the hospitals just couldn't do it. So that's when we started, doing the whole startup thing, taking on investors, like building out the company. So we let another company validate the market for us. Other questions?
I have a question.
Yeah.
Can you talk a little bit about how Carol prepared you for your future, or any advice that you would have for these students in preparing for their own future?
Sure. Well, Carol, from Carol, I got into Yale, so that was really good. And it kind of, my senior year was a bit of a hybrid, because I ended up taking classes at the University of Dayton. And so, and Carol was very flexible with that. So that, for me, that was really interesting, and it allowed me to do something maybe unusual. There was just a lot of flexibility with me being able to follow my interests. And so that was great. My advice would be, and I keep thinking, so I have twin daughters, they're 14. So, I've been thinking, for them for college and all that. And what's really hard is, like, public health was something I would have never normally thought about in high school, because it's just not on the radar. Like, the radar of possibilities is it's just, there's these different paths, and there's sort of the standard things presented, but there's a lot of really, really interesting fields out there, and that kind of combine different aspects of it. It's like, I ended up doing like engineering stuff. I do a lot of communications, a lot of, I do a ton of presentations, a lot of writing, like the patents and the papers and protocols. I do a lot of protocol writing. I've learned, I had to learn microbiology on my own, but it wasn't, so I think that ability to just like be able to dive into anything is hugely important to building a career, because I don't think careers are quite linear anymore. And mine went, I didn't know what I was going to do. I did Peace Corps. I went here, there, just following everything around. But so I guess I just encouraged, like, following, whatever those interests are, and not worrying if it fits a solid path. Because as long as you're doing those things excellent, like the next thing will come, if that makes sense. And then even in college, I would not have encountered public health. It didn't exist on my radar as a thing until I got out. It was actually Peace Corps that kind of had that path. There was like this health path. And normally I would have thought, a health path, that sounds really boring. Because like you think like health class, and like this very basic kind of biology, wellness, but it's totally fascinating, because again, you can go molecular or policy and everything in between. And you can do community, you can do hospital. It's just, there's so much in it. It's like 20% of our gross domestic product is spent on health. So it was a really interesting zone that I wasn't aware of. So I guess what my advice would be is know that those things are out there. There are like things that you maybe haven't figured out that might be really great matches. I don't know. Any other questions, thoughts?
Yes.
Yeah.
How long do you think it's going to take before problematic bacteria and pathogens are going to be resistant or almost near immune to this technology?
Interesting. So I've done some resistance studies on it. So there's multiple ways of creating germicidal light. So they're basically different wavelengths. And what's nice about the pulse xenon system that we have, which is that when you pulse the light, there it is in pharmaceutical. Let me go back to the main page. I have to learn to navigate my own this website. When you pulse the light, it produces what we call broad spectrum or full spectrum energy. So it's producing multiple wavelengths. So 200, 201, 202, 203, 204, produces the entire UVC spectrum. So it gives it multiple mechanisms of damage for an organism. There are other forms of producing germicidal light that are narrow spectrum. So you could take a mercury vapor bulb, apply energy to it, and it will produce the wavelength 253.7 nanometers. You can take an LED, and it'll produce wavelengths around 265 to 270. There's a little bit of a range there. You can take a light at about 400, which is visible blue light, which has some efficacy on, say, MRSA. Now, what we've done to test resistance, because we want to make sure we're not going to create resistance, is it's a very simple experiment. You take a Petri dish, and you inoculate with the bacteria, say E. coli, half the dish, and then you cover it with aluminum foil. And you put it under the different lights. and you just let it grow. You just keep feeding it. It's got all its agar, it can keep growing. And you basically see if it ever grows from under the foil over. So if it never develops resistance, you can take off the foil and you'll have a nice semicircle of bacteria and a semicircle of nice, clean, sterile agar. So when we do that with pulsenon, it works. It doesn't start growing under the light. When we do it with a mercury vapor lamp, because it's only one wavelength, so it's only one mechanism of damage, in about 3 to 4 days, we'll start seeing growth coming out from in the light side, so under the light. When you do it with something like 4 or 5 nanometers, which again has a little bit of a germicidal effect, but it's also a wavelength that bacteria have. It occurs on Earth because it's visible light. It takes about 4 hours to grow. It almost grows at the same rate as, there's only a little bit of suppression of growth, and then that's selected for. And the other thing with resistance that we consider is, because it's only used in the environment, most organisms are in a process of dying off in the environment. So they're not in a place where they're reproducing at such a level that we might see mutations to the system that would cause additional survival, that then would cause them to be more infective and then create an evolutionary pathway. So we're really kind of they will eventually die in the environment on their own, but just not fast enough because the next patient's coming in, 30, 45 minutes after that first patient left. So I think we're pretty good on resistance, but that's a good question. We're starting to do and I'm excited for these, we're starting to do some genetic studies to understand, so we can sequence an organism and understand exactly where the damage we're creating is. But that's a little bit of a back burner project. That's very fun.
So the students have their next class.
Okay, very good. Excellent.
Probably need to wrap it up, but thank you,
Right.
thank you so much for joining.
Oh, happy to do it. Yeah, if you guys think of any other questions, feel free to email me. Happy to answer them.
Okay. Thank you
All
so
right,
much.
cool.
Thank
Yeah,
you.
thank you. Bye-bye.
Bye.